All right, so now we're going to go over our fourth and final learning objective for today's lecture. And just to remind you, I'm sure you should remember this from your action potential lectures, and I believe I've mentioned it a few times too, but the resting membrane potential of neurons is going to be about, on average, about minus 65 millivolts. And when we're talking about that resting membrane potential, it's the potential along the inside of the membrane. So it's on the inside of the cell, right? So here's our postsynaptic neuron right here. And when ion channels open, it's going to allow the movement of ions either into or out of the cell. And the movement of those ions will be determined by the concentration gradient of the ion set up, as well as the voltage gradient present. And if the movement of ions causes the resting membrane potential to become less negative or more positive, so moving in a positive direction, that is called depolarization, and it means that the membrane potential the cell becomes more excitable. Whereas if the movement of ions causes the membrane potential to become more negative or move further away from threshold, called hyperpolarization, then that cell is going to become less excitable or inhibited. And the three major ions that are of concern as far as affecting membrane potential of neurons, it's going to be sodium, potassium, and chloride. Sodium and potassium, these are positively charged cations. Sodium has a higher concentration outside of the cell. Potassium has a higher concentration inside of the cell. And chloride ions are negatively charged ions with a higher concentration outside of the cell. All right, so we have our neurotransmitter being released by our presynaptic neuron. It's going to diffuse across the synaptic cleft and go bind to receptors in the postsynaptic membrane. Now, as a result, we're going to have some postsynaptic potentials happen, and they can either be excitatory or inhibitory. So if the neurotransmitter causes a depolarization on the postsynaptic membrane, that means it's causing it an excitatory postsynaptic potential. If the neurotransmitter causes hyperpolarization on the postsynaptic membrane potential, then that's called an inhibitory postsynaptic potential. Okay, so let's talk a little bit about the excitatory postsynaptic potentials, or EPSPs, first. So here we have our neurotransmitter receptors on the postsynaptic membrane, and are they ionotropic or metabotropic? Hopefully you answered ionotropic. You would be correct, because the binding of the neurotransmitter causes a direct conformational change of these channels and the opening of them so that we can have direct flow of ions into or out of the cell. So in this case, both of these neurotransmitter ionotropic receptors are allowing positively charged ions into the postsynaptic cell. So for example, sodium, because there's a higher concentration of sodium on the outside, so sodium's going to rush in. There's a higher concentration of calcium on the outside, so calcium's also going to rush in. And the influx of these positively charged ions into the postsynaptic membrane is going to cause, so here's our resting membrane potential. We have an influx of sodium and or calcium, and that's going to cause the membrane potential to become more positive. It's going to move closer to threshold, meaning it's depolarizing and it's becoming more excitable. So this would be an EPSP. Now here we have some more ionotropic neurotransmitter receptors, and upon ligand binding, what's happening is, in this first one, positively charged ions are moving out of the cell. So for example, this would be potassium, because potassium has a higher concentration inside. So if a potassium channel opens, it's going to rush out. So we have positively charged ions leaving the cell. Now, with this ionotropic receptor, upon binding the neurotransmitter, negatively charged ions are going to rush into the cell. So this is like chloride channels. Because chloride ions have a higher concentration outside, they're going to rush in. But in either event, whether positively charged ions are leaving or negatively charged ions are coming in, what's happening to the membrane potential is that it's going to become more negative. So here's our resting membrane potential. Those channels open, positive charged positive ions leave or negative ions come in, and the resting membrane potential becomes more negative, or it hyperpolarizes further away from threshold and it becomes less excitable or inhibited. So this would be an inhibitory postsynaptic potential, or an IPSP. Now with this slide, the only thing I want to make you aware of, I don't want you to stress about this slide, is that the fact that if you remember me talking about the synapses and how the majority of the synapses on a postsynaptic membrane postsynaptic cell that we think about are on the axons and the dendrites. But I did mention briefly that there are axo-axonic synapses. So here's our presynaptic terminal, okay, right here. That's where our neurotransmitter vesicles would be. Here's our postsynaptic membrane over here, where our neurotransmitter receptors are. But we have this other neuron with a presynaptic terminal coming down and synapsing on this axon terminal, okay? And what it does is it can release GABA, which is that major small molecule inhibitory neurotransmitter, and it can affect, or it can limit rather, like inhibit how much neurotransmitter gets released by this presynaptic terminal. And so having this presynaptic inhibition can help modulate the activity of the presynaptic neurons and help so it modulates, and it just adds an extra layer of regulation on the presynaptic function. Okay, so it's just an extra layer of regulation and modulation. Okay, so think of it as it allows for more fine-tuning of what's going on. Okay, so don't stress too much about this slide. Just know that that exists. Now, each neuron, every neuron can have, thousands upon thousands of presynaptic neurons synapsing on it, right? So, every neuron can get information from all over. So it's getting a lot of information coming into it. How does it decide? Some of these presynaptic neurons are going to be sending excitatory information. Others are going to be sending inhibitory information. So how does it know what to do? Well, it has to integrate all of that information and decide. And it does that through, so that's called neuronal integration, whenever we have lots of synapses from lots of different areas around the CNS converging on one postsynaptic membrane, postsynaptic cell. But it will decide what to do based off of summation, right? So it's going to add up all of that information, both excitatory and inhibitory, and then what wins out? Is there more excitatory or is there more inhibitory? And that will determine what happens on the postsynaptic membrane. There are two types of summation events that can take place: temporal summation as well as spatial summation. And so you can read through this slide to get if you need better explanation, but I'm going to hit on both of these in the next two slides. So let's talk about spatial summation first, all right? So here, in this little diagram down here, we have the cell body of our postsynaptic neuron, and it's receiving synapses, or it's making synapses with 3 different presynaptic neurons, all right? So this is spatial because it's over the simultaneous release of neurotransmitters from axon terminals from many presynaptic neurons, okay? In this case, they're all sending excitatory information. And so each one of these presynaptic terminals are releasing a neurotransmitter that's going to bind to receptors that causes depolarization on the postsynaptic membrane. And that's going to mean, since it's receiving excitatory information from multiple presynaptic neurons, it's all going to get summed up into a much larger EPSP, and there's a greater likelihood that an action potential will be generated on the postsynaptic neuron, right? So what's happening here, if we look at this diagram or this graph up here, we have our resting membrane potential, and then we have all 3 of those presynaptic axon terminals are releasing excitatory neurotransmitter at the same time. And look, we reach threshold, and now our postsynaptic neuron sends an action potential. But say, for example, only one of these presynaptic axon terminals was releasing neurotransmitter, and these other two weren't. Then it might not be enough depolarization to reach threshold. That concept is shown over here as well. So if we follow the blue line, all right, so this is, we're at resting membrane potential, and only 4 presynaptic neurons fire. And so we get 4 EPSPs, but it's not enough to reach threshold. Green line, we're at resting membrane potential, and we have 8 presynaptic axon terminals releasing neurotransmitter. We get a little bit greater depolarization, but not enough to reach threshold, and it fizzles out. So the EPSP wasn't great enough. But in the case of the red line, we have 16 presynaptic neurons all releasing neurotransmitter, and it's causing so many EPSPs on the postsynaptic neuron that we reach threshold and we get an action potential. So that's an example of summation. This very easily could be all IPSPs, and we could be summing multiple IPSPs at the same time. And then that just means that the postsynaptic membrane would be greatly inhibited, and it would be far less likely that an action potential gets sent. Okay, so the same thing can happen with IPSPs. Temporal summation, what's happening here is here we have our postsynaptic cell body, and we only have 1 presynaptic axon terminal synapsing with our postsynaptic neuron, right? Now, if it only fires once, we might get a little bit. So this is excitatory neurotransmitter causing an EPSP on the postsynaptic cell. And so maybe it only fires once, and we get a little bit of neurotransmitter released, and so we get a little bit of depolarization on the postsynaptic cell. But say that same presynaptic axon terminal fires like 5 times. Well, we get a larger EPSP happening, but still no action potential. Wasn't great enough. But say this presynaptic neuron fires like 100 times, well then we, in a certain given amount of, time period, well now we have an EPSP that's great enough to reach threshold at the axon hillock, and now we get an action potential being fired, right? So not enough EPSP, more firing, a little bit greater depolarization, lots of firing, and now all of a sudden we have a large enough EPSP on the postsynaptic membrane that we get an action potential. The only concept here that I want you to be aware of is that these EPSPs as well as IPSPs, there's a decrement to the excitation and inhibition, meaning that these local potential changes are graded, meaning they start to fizzle out as they spread across the cell body. And so these EPSPs, especially the EPSP, right, you have to have a great enough depolarization that by time it reaches the axon hillock where those voltage-gated sodium channels are, it has to be at threshold by time it reaches here. Don't stress about this slide, okay? And then the only concept that I want to very quickly, that this graph illustrates from the book, is the fact that different neurons can have different thresholds as well as different firing or frequencies of discharge, like how rapidly they can fire action potentials. So here, neuron 1 has the lowest threshold, meaning it's the neuron out of all 3 of these that could be excited the easiest, right? It's the easiest to excite this neuron. Whereas if we look at neuron 3, it has a higher threshold, meaning it's going to be more difficult to excite this neuron because it takes more depolarization to reach threshold, but it has the highest frequency of discharge, so it can fire more action potentials per second than either neuron 1 or neuron 2. Don't stress about the slide. I just want you to be aware that different neurons can have different thresholds and different firing frequencies. And then this, to wrap it up, is just showing you that, or telling you that there are different effects there are different different things that can affect synaptic transmission, like fatigue, pH, hypoxia, and drugs. So just read through this slide on your own, but I wouldn't stress about this as far as an exam question goes, okay? And that's the end of chapter 46.